Our next speaker is Dr. Jessie Guo, resident member of CINJ, associate professor of medicine at the Rutgers Robert Wood Johnson Medical School. Jessie received her PhD from Duke University and did postdoctoral training at CINJ. The title of her presentation is Role of Autophagy in KRAS-driven lung cancer. Jessie.
Okay, yeah, thanks introduction from Nat. And actually, so my lab have been interested to understand the role of autophagy in KRAS-driven lung cancer for years. And as we know that autophagy, in the United States, among the top 10 leading cancer types, the incidence rate of lung cancer is second highest in both men and women, but the mortality rate is very highest. And in my lab, we are mainly interested in understanding the role of autophagy in the non-small cell lung cancer, which includes adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. And the somatic mutation has been observed in the lung cancer. So among them, like TP53, KRAS, and STK11 are the three most frequently mutated genes have been detected in the non-small cell lung cancer. And also here is the study from the MR expression in the KRAS-driven lung cancer from human cancer patient. They found that indeed the KRAS-driven lung cancer can be divided by the three clusters. When they check with the genetic mutation status, and these three clusters belong to the KRAS mutation alone, or co-mutation of KRAS with LKB1, or the co-mutations of KRAS with TP53. Indeed, many preclinical mouse studies and also clinical studies have found that those different subtypes of KRAS-driven lung cancer, they have different biology, different gene expression, different immune profiling in the tumor microenvironment, also different metabolic vulnerability, and which result to the different response to the standard cancer treatment. So we know that the cancer cell reprogram the metabolism for survive the metabolic stress microenvironment, like the KRAS activation have been reported to upregulate the glucose glutamate uptake, promote macropinocytosis, also upregulate the autophagy. So my lab mainly interested in understand how the autophagy is essential or support the KRAS-driven lung cancer. So briefly introduce autophagy. So autophagy is a process involved in the protein and the organelle degradation. So in nutrient-rich condition, autophagy is inhibited by the mTOR complex 1. During metabolic stress, autophagy activation and begin with a double membrane called FAG4. Then this double membrane elongation and engulf the protein aggregates, damage the organelles and other cytoplasmic components to form autophagism. Then autophagism fills the lysosome to become autophagolysosome and degrades cargos and release the building block to the cytoplasm. So in general, we believe that autophagy has two major functions. One is the organelle and the protein quality control function. The other is intracellular cycling function. Indeed, the cancer cell utilize this function to maintain the energy homeostasis, to maintain the nucleotide pool for the survival and the proliferation. So actually, the autophagy machinery is very complicated. As I said, during metabolic stress, autophagy will be activated through the AMPK activation, which can directly or indirectly activate the ULK1 complex, which is the pre-initiation complex. Then this complex will promote the initial complex formation, which can promote the autophagism formation, maturation, and finally become the autophagism. So indeed, during the autophagism maturation processing, so there are two ubiquitin systems that are involved. One is the ATG5-ATG12 conjugation, which is mediated by the ATG7E1-like enzyme. Then they form a complex with ATG16. This will promote the LC3 cleavage and processing to then ATG7 be involved for the LC32 lipidation. Then this LC32 lipidation, lipided LC32 will bind to the autophagism inner and outer membrane. And then another protein called the P62 is a cargo protein, adapter protein, then can deliver those cargoes to bind to the LC32 and then form the matured autophagism. Then this autophagism will fuse lysosome to become autolysosome and then degrade cargoes and recycle these building blocks for cell to survive. So in my lab, yeah, we are interested to know how the autophagy support KRAS-driven lung cancer using the genetic engineered mice models. As I said, so the KRAS-driven lung cancer can be divided by the 3 different subgroups of KRAS-driven lung cancer. So we generate the gems with the ATG7 in the wild type of flux with the either KRAS mutation alone or KRAS mutation with the p50 flux or KRAS mutation with the LKB1 flux. So then we can intranasally infect those mice with the lenticule to induce lung tumor. Meanwhile, we can delete the autophagy essential gene 87. They can understand the role of autophagy in the tumor growth. And here I'm going to show the one example we've found is that here we can see in the mice bearing the ATG7 KL lung tumor. Over time course study, the lung gradual increase indicates more tumor growth. But if you delete the ATG7, indeed those mouse bearing ATG7 deficient KL lung tumor, so we don't see the lung weight increase. And we further check the histology by the H staining and the quantification of tumor burden and tumor number and tumor burden. So we can see that ATG7 deficiency can significantly inhibit the KL lung tumor initiation and show reduced the tumor number and also the further tumor growth. So inconsistent with the reduced tumor growth, we also found that the mice bearing ATG7 deficient KL lung tumor significantly extend mouse lifespan by more than 2 months. And indeed, the same effect we also found in the KP lung tumor model. So we do see that ATG7 deficiency also inhibit the KP tumor growth. And here I'm just show the survival curve study. You can see also in the mice bearing the ATG7 KP, ATG7 deficient KP tumor can significantly extend mouse lifespan compared to the mice bearing ATG7 wild type KP lung tumor. So from our gem models, we're demonstrating that tumor cell autonomous autophagy is essential to support the KRAS-driven lung tumor growth. And we further explore the underlying mechanism. I'm not going to show detailed data, but just a model show over here. We do found that tumor cell utilize autophagy to degrade those macromolecular then to provide the amino acid, which are essential to maintain the tumor cell TCA cycle to maintain the energy for the energy production, like ATP production. And meanwhile, also the autophagy provide those to provide an acid is sufficient to maintain the nucleotide pool, which are essential for the tumor to survive those metabolic stress. So from our current gem model, we're using the flux cream model. Actually, so indeed, so far we know that tumor cell utilize autophagy for them to survive, and which tell us targeting autophagy could be a good strategy for treating the KRAS lung cancer. However, the one caveat for this model is that we delete autophagy the same time as the induced oncogenic event, which really doesn't mimic what's going to be happened in the clinical practice when we're using the autophagy inhibitor. So to avoid this limitation, so we generate another mouse model as autophagy inducible mouse model. So in which we can temporarily control the autophagy essential gene ATG5 expression by using tap regulated shRNA against the ATG5. So in this study, so we can easily specifically knock down ATG5 in the tumor, or we can systematically knock down ATG5 in the tumor, in the host. So what we did is first we introduced lung tumor, and then we give a mice with doxycycline. Then we can check how the tumor specific knockdown effect on tumor growth or the systematic ATG5 knockdown on the tumor growth. Here, just Western blot to show that we can successfully knock down the ATG5, either tumor specifically, which show reduce the ATG5-12 conjugation and also increase LCD1 accumulated indicate autophagy blockade in the tumor. And also here is one model, systemic ATG5 knockdown model. We can see that ATG5 have been knocked down in the multiple tissues, also cause accumulation of LCD1. So once we have established the mouse model, so we began to understand the effect of the role of tumor intrinsic autophagy or the system on the established KRAS-driven lung tumor growth. And here we are using the KP model to show. So we induce the KP tumor first. And when tumor established, we give the mice with doxycycline diet and then study time course study to check the tumor growth. And unexpectedly, indeed, when we have the KP tumor ready, then we get knock down ATG5 in the tumor. We didn't see any tumor reduction. But if we systematically knock down ATG5, so we can see the systematic ATG5 knockdown significantly inhibit the KP lung tumor growth. Here show lung weight and tumor burden analysis. Also, histology can see the clear effect. So next, we want to know what the mechanism, how the system autophagy support established KP lung tumor growth. As we know that autophagy essential, as I introduced before, so autophagy essential to maintain the TCA cycle metabolism for the energy production. And George also talked about a lot background to show how the TCA cycle glycolysis is essential for the ATP production. I'm not going to more, but what we did actually is that we used the C13 uniformly C13 label glucose for the in vitro study in the mice, either ATG5 wild type or ATG5 knockdown. So here, so first we do see that in the system ATG5 knockdown, so the glucose carbon flux go to the tumor TCA cycle metabolites was significantly reduced. Here's total enrichment. Also, we can see the M+2 is also significantly dropped in the system ATG5 knockdown mice. But this effect we didn't observe in the tumor-specific ATG5 knockdown tumors. And also we checked the ATP level. We do see that system ATG5 knockdown can significantly reduce the ATP level in the KP lung tumors. So next, actually, so we know that the in addition to glucose contribute carbon for the TC cycle metabolite, and the glycolysis, indeed, glucose through glycolysis contribute those carbon for the biosynthesis. So also then we track the glucose carbon to the glycolysis intermediate, also go to the serum. So we do see here the result to show that in the system ATG5 knockdown mice, the glucose carbon flux to the G6P lactate is significantly lower, and indicate the reduced glycolysis. And also we do see the significantly reduced glucose flux go to the serum, suggesting that also the serum biosynthesis is impaired by the system ATG5 knockdown. But this effect is not observed in the tumor-specific ATG5 knockdown tumors. So next, indeed, we did another tracing study selected, as I think Josh already said, that introduced a very good background for this study. So glucose lactate, another carbon source for the tumor TC cycle metabolites. So that's why, so we did the lactate tracing study in our mice after the 4 weeks doxycycline treatment. So here, so lactate contributed pyruvate and go to the TC cycle, support the go to TC cycle. But we, in our tracing study, so we do see that in the system ATG5 knockdown mice, there's less lactate uptake in the tumor and reduce the lactate carbon go to the pyruvate. But the lactate carbon flux go to a tumor TC cycle is not significantly different, although they're slightly reduced trends. Another interesting data we observed is that we have the glucose, the lactate carbon flux go to the glycolytic intermediate is significantly lower. So here like the G6P and F6P. So it has been reported in many studies to showing that cancer cell indeed hijack the glucogenic enzymes to engage in a truncated glucogenesis, then which can be used for the biosynthesis or for the antioxidant defense. So actually in our tracing study suggesting that in the, so actually autophagy might be essential to support the tumor glucogenesis, which might be used for the serum biosynthesis. Here we do see that the carbon flux from lactate to the serum is also significantly reduced in the system ATG5 knockdown mice. But these phenotypes are only observed in the tumor, but we don't see it in the liver or other tissue, although this model is a system ATG5 knockdown. So from here, based on by those in vivo isotope tracing study, we demonstrated that system autophagy is essential to support the tumor cell metabolism, which is one of the mechanisms to support tumor growth. Actually, also another study, including Dr. White lab studies, have demonstrating that host autophagy is also essential to support the tumor growth by the autophagy mediated the hepatic autophagy immunotolerance. So if you inhibit host autophagy, you inhibit the T cell mediated tumor killing. So then we want to know if really the host autophagy mediated anti-tumor immune response is also essential to inhibit the established KP lung tumor growth. So to do this, we first checked the tumor immunoprofiling in the tumor microenvironment in the mice treated with doxycycline for 4 weeks. And over here, we can see that in the system ATG5 knockdown mice, the CD4, CD8 T cells and the CD4 memory T cells significantly increased in the KP lung tumor microenvironment. However, if you only knock down ATG5 in the tumor, this has no effect on the tumor cell infiltration, indicating this the host effect, not the tumor specific effect. So next, we want to know what is the consequence of those increased tumor infiltration in the KP lung tumor microenvironment by the systemic ATG5 knockdown. So to do this, we first induce a lung tumor, and then tumor established. We use the anti-CD4 and the CD8 antibody to co-deplete the CD4, CD8 T cells. Meanwhile, we knock down autophagy, systematically give the mice with doxycycline treatment. And then after 3 weeks, we monitor the tumor growth. And first, as in the IgG control group, as we expected, system ATG5 knockdown significantly inhibit the established KP lung tumor growth. However, if we knock down the CD4 and deplete CD4 and CD8, so this effect was totally disappeared, which suggesting that system autophagy might also suppress the T cell mediated tumor killing for the established KP lung tumor growth. So advantage for this mouse models is when we stop doxycycline treatment, we can restore the autophagy. So therefore, after knockdown autophagy for a while, we have know that we can inhibit established KP lung tumor growth. We ask if we stopped autophagy, restart autophagy, what happened for tumor growth. So here we stop the doxycycline for 3 weeks and select the mice to check the tumor growth. And here the good news is that even with short-termly restored autophagy, so the tumor didn't grow back, suggesting that transient system autophagy indeed can irreversibly inhibit the established KP lung tumor growth for short term. Meanwhile, we also checked the infiltration in those tumors after we stop the dox treatment. Actually, we do see even when autophagy is restored, and we still can see increased immune cell infiltration in those tumors, suggesting that maybe the tumor didn't grow because we still have the anti-tumor immune response happening in the tumor microenvironment. So another study we did is actually after we ATG5 restored, we also performed the isotope tracing with the lactate. So we do see that even with autophagy is restored in the short term for 3 weeks, and the one thing is flux from lactate to the pyruvate still has been reduced. And also in this case, we also observe that flux from lactate to the tumor TC cycle is also significantly lower, even autophagy is restored. Another thing we do see that, as I introduced before, so we do see the system ATG5 knockdown can impair the tumor glucogenesis. Even with restored autophagy for short term, this impaired tumor glucogenesis still can be maintained. And also the impaired the serum biosynthesis still maintained when we restored autophagy for the 3 weeks. Actually, then this good news suggesting that even autophagy restored and there's the tumor or tumor environment, maybe still have memory to maintain the anti-tumor response. So as we know that besides the autophagy essential for tumor growth, the normal tissue is also requires autophagy to maintain the homeostasis. And we have to know that when we see the knockdown autophagy, one clear phenotype we can see is increased liver weight and also increased spleen weight, because liver essential requires autophagy to maintain the function. And but if we restore the autophagy, so this increased liver weight and the spleen weight can quickly recover to normal. Another phenotype we observed is that the system autophagy knockdown caused about 5 to 10% mice have the ulcerated skin. So, but if we ceasing dogs for 2 weeks, this ulcerated skin problem also can be restored. So compared to the effect of tumor with autophagy restoration, indeed, the restored autophagy, we can recover the normal tissue damage, but can still inhibit the tumor growth, demonstrating that there is a therapeutic window we can use to target autophagy for the cancer treatment. So finally, we mimicked what's going to be in the clinical practice when we use the autophagy inhibitor. So we intermittently treat the tumor-bearing mice with doxycycline to 10 on, 10 off autophagy cyclically. So we do see that cycling transit system ATG5 knockdown and restoration can significantly extend the mouse lifespan compared to the autophagy wild-type mice. So in addition for the KP tumor, so we also generate this same system to test if this fact can be observed in the KL tumor, and indeed we do. Even when we have a KL tumor first, when we the intermittent knockdown AG5, we can significantly extend mouse lifespan. But another interesting observation, we also found that in the KL tumor, even with tumor-specific knockdown autophagy, we can also significantly extend mouse lifespan. So in summary, using the autophagy inducible mouse model, so we do find that the system autophagy is essential for the established care-driven lung tumor growth. The potential mechanism may be that the one thing through T-cell mediated tumor killing, and also system autophagy might support the biosynthesis and also essential for the energy production. Then all this is essential for the tumor growth. But if we inhibit the system autophagy, which can inhibit the increased anti-T-cell mediated tumor killing and also prevent the inhibit tumor metabolism, which cause the tumor reduction. And most importantly, is that if we short-term recover the restored autophagy systematically, indeed this anti-tumor effect still can be sustained by the T-cell mediated tumor killing or maintain the anti-tumor metabolism. So our study is suggesting that really we can target in the autophagy as no strategy for the care-driven lung cancer. However, autophagy inhibitor alone cannot be used cancer treatment. Usually it promotes to combine with other treatments. So in our study, so we are proposed to use the autophagy inhibitor combined with the MEP inhibitor, because targeted therapy so far is not really successful in treating the care-driven lung cancer, except for the G12C inhibitor. So in this study, so we propose to use the hydroxychloroquine, which is a FDA-approved autophagy non-specific inhibitor with a trametinib. So we use the different mice models we had in the lab, either allograft model or the GEM model, also PDX model. So I'm going to show some data we got so far. So one thing data we did is we had large generate tumor derived cell lines. So we just allograft inject in the nude mice and then treat mice with trametinib alone, or hydroxychloroquine or the combination. So here's the KL allograft tumor growth. We can see that hydroxychloroquine combined with the trametinib can significantly inhibit the KL allograft tumor growth. And also you can see the final second mice tumor weight is significantly lower in the combination treatment compared to the control or the single treatment. However, when we use the same concentration of drug to treat the KP allograft tumor, indeed we couldn't observe this anti-tumor effect by the combination treatment. So this, except for nude mice allograft tumor growth, also we validated allograft tumor growth using the 67 syngeneic mice model. So same thing, we observed that combination treatment can only inhibit KL allograft tumor growth, but not KP allograft tumor growth. So next, we use the GEM model, which although take time to do study, but we still finally get this clear result. So what we did is we introduced the KL or KP lung tumor in the mice first. When tumor established, and we treat the mice with the single treatment or the combination treatment. And over here, we can see maybe this one much more clear. So we can see compared to the single treatment or the vehicle control, combination of hydroxychloroquine with MEK inhibitor can significantly inhibit the KL lung tumor growth. And also we have the quantification of the tumor number, tumor burden. We do see that the combination therapy can inhibit the KL lung tumor growth. But when we do the same effect, the same concept drug, in the KP GEM model, indeed we don't see any anti-tumor effect by combining with hydroxychloroquine with the MEK inhibitor. So finally, we also use the PDX model. So we got from the Jack's lab, which one for KL PDX model has the co-mutations of KRAS and LKB1 and KP with co-mutation of KRAS with TP53. So from here, so we do see that combination of hydroxychloroquine and the trametinib can significantly inhibit the KL allograft tumor growth compared to the single drug treatment or vehicle control. And here just show the tumor weight, we can see that clear effect. But for the KP PDX, although we can see trametinib can inhibit the tumor growth, but the synergistic effect, we couldn't see that combination has the much more anti-tumor effect. So from those mouse in vivo studies, so we do observe that combination with autophagy inhibition with trametinib can only specifically inhibit the KL lung tumor growth, but not KP lung tumor growth. Next question we would like to know is what is the mechanism for the combination can inhibit the KL tumor growth? We know that autophagy is essential to inhibit stress to prevent ROS level. So if we inhibit autophagy, which can cause the increased ROS and also might increase lipid peroxidation. And also another study has been shown that ROS activation indeed inhibit fructosis. So we are thinking that maybe because we know that lipid peroxidation also can promote the fructosis. So we hypothesize that combination with hydroxychloroquine blocks autophagy or the MEK inhibitor to inhibit the MEK might be increase the fructosis, which can cause the reduced tumor growth. So we did a lot of in vitro culture study. I'm not going to show data, but here's our in vivo study. So we do see we are using the lipostatin, the one which is the fructosis inhibitor can be used for in vivo. So here's the allograft tumor growth. And in the left side, we can see the in vitro control group. So the combination treatment can significantly inhibit the KL allograft tumor growth. But if we give the lipostatin 1, so they can successfully rescue this reduced tumor growth, which demonstration definitely combination of hydroxychloroquine with trametinib induced the apoptotic cell death. So the last part in summary, so we do use the different in vivo tumor model demonstrating that combination with autophagy inhibition, hydroxychloroquine with trametinib can specifically inhibit KL lung tumor growth, which might be through the induced apoptotic cell death. So finally, I would like to thank the people from my lab, especially most work I produced today from the previous postdoc Kushi and the previous graduate student Ru Shang, also Taijin provide for the KL inducible mouse model, and also our collaborators, especially Sherry, helping do a lot of, from my lab, helping do a lot of the surgery, allow us to do the tracer study, and also the Dr. Xie helping with immunoprofiling, and Xiaoyang helping a lot of metabolomics, the running data, and Sharon helping with PDX model, and also the Dr. Masashi, the narrator from the cancer research at UK Cambridge, indeed provide us inducible mouse model, which allow us to cross-breeding with RKP model to study the systemic tumor-specific knockdown for the autophagy. And also, of course, always George Business Lab helping a lot of the suggestion of for the tracing study. So I would like to also my funding agencies. I'm going to stop here. So